Alright, so we're going to talk about genes and variations, and this is really microevolution. So we're talking about changes in gene frequency and allele frequency in a population over time. When we look at genetics, we're talking about variations, so we're talking about those differences in a population. They don't necessarily make survival better or worse, but they're just inherited traits that are natural, and they are basically what is then used to determine things for natural selection. Just as a reminder, important words like genotype, that's the combination of allele, phenotype, that's the physiological, behavioral characteristics. Natural selection acts on the phenotype, not the genotype. And so organisms that with a higher fitness, they're the better suited phenotype. And so then they'll pass on their genetic information, but natural selection acts on that phenotype. Some other important words, so we have population, that's a group of individuals of the same species that mate and produce offspring. We talked about species before, and so these are going to be organisms that are going to be capable of reproduction and making fertile offspring. We have a gene pool, so this is going to be all of the different alleles for each gene that are present in a population. And then we have the allele frequency, which is the number of the time that the allele occurs in a gene pool compared to the total number of alleles. And when we look at microevolution, we're looking really at the change in this allele frequency. And so it's the change in that frequency of alleles over time. Remember that populations evolve and not individuals, and so natural selection might operate on the individual organism, so an organism with an adaptation that is beneficial will survive, but the change in allele frequency is what we measure inside of a population. An example of allele frequency would be something like this, and so we're looking at the trait of fur color in mice. So mice that are dark are going to either be homozygous black or heterozygous black, and then the brown fur will be homozygous recessive, and so the frequency with which the alleles occur is shown to the right, and so that heterozygous, even though it has the black phenotype, it still has that allele for brown fur, and so that allele still exists, and so we can still have brown mice. Whenever we look at sources of genetic variation, we look at three sources: mutations, genetic recombination, and lateral gene transfer. So mutations we talked about in genetics, and meiosis, we're looking at the change in the genetic material, change in nucleotides, change in chromosomes. If it's something like a silent mutation, it's going to be neutral, it won't change the phenotype, but if the mutation impacts the fitness of the organism and it will change the phenotype. Whenever we look at mutations, each person is born with somewhere around 300 mutations that makes their DNA different, but most of them are neutral. There might be a few harmful, a few beneficial, but in order for mutations to actually make a difference in evolution, then they need to impact the cells that make, the germ cells that make eggs and sperm. If they're just happening in your somatic cells, you're not going to pass that on to your offspring, so in terms of evolution, it doesn't necessarily matter. Another form of variation is going to be genetic recombination, and whenever we look at genetic recombination, we're talking about basically independent assortment, which we looked at with meiosis, and then crossing over, which we looked at with meiosis as well. And so this is new combinations of traits, and this would be a natural source of genetic variation within a population. We can also talk a little bit about lateral gene transfer, and so this is going to be passing genes between individuals, not offspring. So they're going to be in the same generation. This happens in bacteria. They trade plasmids, which are basically circles of DNA, and they trade them back and forth, and this is where a lot of the antibiotic resistance that we see come in. Quickly back to mutations, just a few thoughts about how they can be harmful, neutral, or beneficial. So if it doesn't change the phenotype, then there will be no effect. So that's a silent mutation. Or if it doesn't change the fitness, then it will be no effect. If the mutation alters the phenotype, then it can either be harmful if it lowers the fitness, or it can be beneficial if it raises the fitness or makes it more fit. When we look at how natural selection impacts different traits, we can look at single gene traits or polygenic. So single gene traits, meaning a trait just controlled by one gene. This is going to, whenever natural selection acts, it's going to change allele frequencies and therefore change the phenotypic frequencies. So we're going to see different amounts of each different phenotype for that organism. For a polygenic trait is going to be a trait that is controlled by many genes. And whenever we look at this, we kind of look at it as a bell curve. And we look at, so maybe normally it's a standard bell curve where most of the population is kind of in the middle with the phenotype, and that's kind of how it looks. But we have stabilizing selection, directional selection, disruptive selection as two different kind of pictures of how natural selection acts on polygenic traits. So stabilizing selection means that individuals at the center of the curve have higher fitness than those at either end, and so the curve narrows overall. So for example, babies that have an average birth mass are more likely to survive. So overall, the curve just kind of gets narrower and pinches into the middle. For directional selection, what we're looking at with that is we're looking at our phenotypes where an individual on one side of the curve has better fitness than the other. And so if we were going from beak size small to large, this one would be an example of, oh, well, larger beak size helps the bird to survive more. And so the population in general would shift to having a larger beak size that is larger than usual. If we have disruptive selection, then it means that individuals at either end of the curve have a higher fitness than those in the middle. And so it eliminates that medium or kind of the in-between phenotype. So if we're looking at birds and their beak size, if we have kind of a range between small and large, if that medium one doesn't have the ability to survive as well, then over time you'll see actually two populations emerging where you have a population that has a really small beak and a population with a larger beak. And this over time can really lead to speciation and can lead to the formation of that new species. So the last thing we'll talk about is another type of evolution which isn't really dependent on traits. This is called genetic drift. This is going to change the allele frequency, but it's going to change it because of chance rather than natural selection. Genetic drift occurs whenever there's a small population. If certain individuals migrate outside of the population, so they leave, and then over time, if there's different occurrences that happen, then they can cause a certain allele to become more or less common in a population. So there could be environmental catastrophes, things like that, that also contribute to this genetic drift. There are two types of genetic drift that we'll look at. So we have genetic bottlenecks, which is a change in the allele frequency that is followed by follows a dramatic reduction in the size of the population. So there's something that happens, the population goes from 1,000 to 100, and because of that, you have a really good chance that you're going to change the allele frequency, especially if it was just some sort of random event, like a natural disaster. Then you're going to have a small population, and now that population only has certain alleles that it's going to be able to pass on to the offspring. The other type of genetic drift is the founder effect, and this is going to be a decrease in genetic variation that's caused by a new population forming and a migration, essentially, of a small part of the population. So a small part of the population migrates, either it's by chance, and so that population will have a different frequency of alleles than the original, and so then over time they will become likely much more different, and this could also lead then to changes in that allele frequency and maybe even speciation over time. With genetic drift and the founder effect and bottleneck effect, this is really where we could find some rapid evolution, so that punctuated equilibrium is that some drastic change, natural disaster wipes out, 90% of the population, that's going to lead to a fast change in that population in terms of evolution compared to natural selection, which is going to probably gradually change it, and you're going to see over time that the organisms that have the best traits survive. With genetic drift, this is by chance, this is random, this is going to be something that can cause a really fast change in a short period of time. Whenever we look at, so we've been talking about evolution and change over time, whenever a population reaches genetic equilibrium, this means that the allele frequencies aren't changing, so no evolution is occurring.